Welcome to the Innate Immunity module, and in this talk we're going to focus on the classical pathway of complement activation. We will learn about how the classical pathway is activated as well as the outcomes of the classical pathway. The complement proteins are a collection of more than 30 different soluble proteins that are present in blood as well as bodily fluids. They were initially discovered in vertebrates. However, now we know that homologs of some complement proteins are also found in invertebrate organisms. A lot of complement proteins are thioester proteins, and when we look at vertebrates, the complement proteins are mainly synthesized in the liver. Now, the complement proteins work together and form a cascade that's called as the complement pathway. The complement pathway aids in the pathogen elimination as well as induction of inflammation. These complement proteins on their own are not antimicrobial in nature, but when the complement pathway gets activated, that is when pathogens can get eliminated. Now, when a pathogen is able to overcome the epithelial barriers of the host and enter the host, that is when the complement system is activated. There are three main pathways of complement activation that have been studied in vertebrates. The first one is called as the alternative pathway. The second one is the lectin pathway. And then we have the classical pathway. Now, the complement system is an ancient form of host defense. And so when we look at the evolution of the complement pathways, the alternative pathway evolved first, followed by the lectin pathway, and then the classical pathway. The classical pathway involves the complement protein C1, which upon activation is able to cleave two complement proteins called C4 and C2 that result in the formation of an enzyme called the C3 convertase. The C3 convertase is able to cleave another complement protein called C3. And the result of that is the formation of another enzymatic complex called as the C5 convertase. The C5 convertase is able to cleave the protein C5, and one of the products of that cleavage, which is C5b, is able to initiate the terminal part of the classical pathway. The classical pathway of complement activation was the first complement pathway that was discovered. The pathway is activated when the complement protein named C1 binds to either a pathogen or an antibody that is bound to the pathogen. It is important to note that since the C1 protein is able to recognize and bind to antibodies that are bound to pathogens, as shown in this figure, this pathway functions as a bridge between innate and adaptive immunity. The classical pathway is activated when the complement protein C1 is able to bind to a pathogen or an antibody that is bound to the pathogen. Now, C1 is made up of three subunits. One is called as C1q, another one is C1r, and the third one is C1s. Now, in the case of the complement C1, the C1q subunit is similar to the mannose binding lectin and the ficolin when we look at its structure. On the other hand, C1r and C1s are similar to the MASP proteins that associate with MBL and ficolin. Thus, there are structural similarities in the composition of the C1 complement protein along with the MBL and ficolin that have MASPs associated with them. When the C1q part of the C1 protein binds to a pathogen or antibodies that are bound to the pathogen, it activates the protease C1r, which will then activate the protease C1s. The C1s protease then interacts with the complement protein called C4, and it cleaves this protein into two fragments, the smaller C4a fragment and the larger C4b fragment. The C4b fragment is able to form a covalent bond with the proteins that are on the cell surface of that pathogen. because the C4b fragment has a reactive thioester bond. This reactive thioester bond can react with hydroxyl or amino groups that are present on proteins of the cell membrane of the pathogen. And by forming this covalent bond, it is able to bind to the cell surface and coat the cell surface of the pathogen. This reaction has to occur very fast, and if this reaction does not happen quickly, then that thioester bond is hydrolyzed by reacting with the surrounding water. And when this happens, then the C4b fragment will not be able to covalently bind to the cell surface of the pathogen. In addition to C4 cleavage to form C4a and C4b that is able to bind to the surface of the pathogen, C1s also can interact with another complement protein called C2. And this interaction results in the cleavage of C2 into a smaller fragment called C2b and a larger fragment called C2a. The C2a fragment is able to interact with the C4b fragment that was generated through the cleavage of C4, and C2a and C4b can interact with one another to form a complex that's called as the C4b2a complex, which has enzymatic activity and is also called as the C3 convertase. Now C3, which is cleaved by the C4b-C2a complex, which is also called as the C3 convertase, it generates a smaller fragment called as C3a, which is a soluble molecule. So the C3a does not bind to the surface of the pathogen, but on the other hand, it can diffuse because it's soluble, and it is an anaphylatoxin. An anaphylatoxin is a molecule that is able to promote acute inflammation. Now, C3a is able to promote acute inflammation by enabling chemotaxis to occur, increasing permeability of blood vessels, and activating immune cells. The C3b fragment that is formed, on the other hand, is deposited on the pathogen's surface because the C3b molecule has a reactive thioester bond similar to the C4b molecule. And this reactive thioester can covalently bind to hydroxyl or amino groups that can be present on the surface of the pathogen. Thus, the pathogen's surface is coated by C3b, and C3b functions as an opsonin. And opsonin is an agent that promotes opsonization, which is another name for phagocytosis. So immune cells that have complement receptors that recognize C3b can phagocytose the pathogen that is coated with C3b, and thus it can destroy the pathogen. The C3b that is formed can also bind to C4b2a, which is the C3 convertase, to form another complex called C4b2a3b. And this complex also has enzymatic activity and is called as the C5 convertase. This C5 convertase now has the ability to cleave the complement protein C5 into two different fragments. One of the fragment that is generated, which is the smaller fragment, is C5a. Now C5a, like C3a, is a soluble and diffusible protein fragment, and it functions as an anaphylatoxin. The anaphylatoxin is able to promote acute inflammation by increasing the permeability of blood vessels, by also promoting chemotaxis of immune cells to the site of infection, and by activating some immune cells. The other fragment that is generated by the cleavage of C5 is C5b. And C5b is able to initiate the late or terminal events of complement activation, which is the formation of a membrane attack complex, which forms a pore-like structure on the phospholipid bilayer of the pathogen, and thereby it promotes cell lysis and killing of the pathogen. Now let us see how the membrane attack complex is formed. Once C5b is generated, it is able to bind to another complement protein called C6. The complex of C5b6 is able to recruit another complement protein called C7, and the interaction of C7 with C5b6 is able to expose a hydrophobic region on C7, and this allows the C5b6-7 complex to be on the plasma membrane of the pathogen. They recruit another complement protein called C8, and thus we form the complex of C5b, 6, 7, and 8. This complex can recruit another complement protein called C9 to form another complex. But the neat thing is it doesn't recruit only one C9 molecule. It can recruit many C9 molecules, anywhere from 10 to 16 of them. And these C9 molecules arrange themselves in a ring structure to form the membrane attack complex. The membrane attack complex forms a channel-like structure in that phospholipid bilayer of the pathogen, and this pore-like structure is able to disrupt the proton gradient across the membrane of the cell because it allows the flow of different ions and other substances between the cell and the environment. The membrane attack complex also allows water and enzymes like lysozyme to enter the cell from the environment, and thus the cellular integrity is compromised, which results in the death of that cell. With this, we come to the end of the classical pathway, where the complement protein called C1, which is made up of C1q, C1r, and C1s, is activated when it binds to a pathogen or an antibody that is bound to a pathogen. Upon activation, the C1 protein is able to interact with two complement proteins, C4 and C2, and is able to mediate the proteolytic cleavage. The result of that proteolytic cleavage is the formation of the fragments C4a and C4b, as well as C2a and C2b. Now the fragments C4b and C2a interact with one another to form an enzyme called as the C3 convertase. The C4b2a or the C3 convertase will interact with the complement protein called C3 and cleave it into two fragments. The smaller fragment or C3a is diffusible and soluble, and it functions as an anaphylatoxin to induce inflammation. The larger fragment, which is C3b, covalently binds to the surface of the pathogen and is able to act as an opsonin, where it mediates the engulfment of the pathogen by immune cells through the process of phagocytosis. Another function of C3b is to be able to interact with the C3 convertase to form a complex called as C4b2a3b, which is also called as a C5 convertase. The C5 convertase will interact with the complement protein C5 and cleave it to a smaller fragment called C5a, which is soluble and hence can diffuse away and induce inflammation because it is a potent anaphylatoxin. The larger fragment C5b is able to recruit other complement proteins that include complement protein 6 all the way to complement protein C9, and these proteins come together to form a membrane attack complex that will lead to the elimination of the pathogen. With this, we come to the end of our talk, where we learned about the activation of the classical pathway and how the classical pathway results in the elimination of the pathogen as well as the induction of inflammation.